Okay. Great. So for anyone who doesn't know me, my name is Megan Slocum. I use she, her pronouns, and I am the current Environmental Science and Engineering Fellow at the Neponset River Watershed Association. One of my main responsibilities as the fellow is to help organize our community water monitoring network. And part of that is running this little report every year. So in terms of how this meeting is going to work tonight, got a little agenda here. The first 45 minutes or so are going to be spent on our 2021 water quality report. Start with a little introduction to the Neponset River Watershed Association and the Community Water Monitoring Network itself. Then we'll transition into some of the actual results. And I'll finish my presentation by introducing some of the really exciting projects that we have going on in the watershed. And then the last 15 minutes of our presentation is going to be, I'm going to kind of give control over to Sean McCanty. Sean McCanty is our new River Restoration Director. We're super excited to have him. He's been fantastic so far, and I'm sure we'll continue to blow it out of the water. He's going to talk a little bit about his PhD research and how that's led him to becoming the River Restoration Director here with NepRWA. Okay, so we're going to start with some background on NepRWA and the Community Water Monitoring Network. From here on out, I'm going to refer to it as SWMN, which is our fancy acronym that we use. And that's just talking about our community science project we use to collect water quality data. So the Neponset River Watershed Association is a grassroots nonprofit based out of Canton, Massachusetts. So you can see here's a map of Eastern Massachusetts, and I've outlined the Neponset River Watershed. So we have just a little bit of Boston, but most of us is just south of Boston. The Neponset River Watershed Association, our main goal is to really increase the health of and access to the Neponset River, its tributaries, and the surrounding green spaces. And we do this through multiple different avenues. So we work on increasing access through public events that allow people to get out on the water and paddling. We also do a lot of advocacy, which helps to protect green spaces and our rivers. Then we also have ecological restoration projects we work on. This is drone footage of one of our current projects over at Mill Pond. This pond was originally created to help increase the amount of space for, swimming and fishing. But over time, it's been filled with sediment, which you can see here. And now we're trying to work to increase the habitat and make it healthier for things like fish and macroinvertebrates by taking out the dam that's created the pond. We also do a lot of work with education. Here's a picture of one of our more formal forms of education. We go into 5th grade classrooms. We also go into high schools and we talk about water conservation and stormwater runoff. We also do education that's a little less traditional. So we work with homeowners to make sure that they know how they can be good stewards of the Neponset River watershed. And then finally, we also focus on water quality. And that's going to be the main story I'm telling tonight. We focus on water quality primarily through our community water monitoring network. So our SWMN program is this really fantastic community science project. We have 41 sites in the Neponset River watershed that we sample. So this is a map here on the left with the Neponset River watershed outlined, and then each of those dots represents one of our different sampling locations. And we have pretty good diversity. We have sites on the main stem, we have sites on tributaries, and we also have a few on different ponds and lakes. And these sites are sampled 6 times a year, monthly from May through October. And each of these sampling events involves over 50 volunteers. And we're really doing this to monitor our water quality. So this program's been going on since the mid-1990s. That means that we can look at long-term trends at our sites, and we can also flag sites where there are potential issues happening. And one of the main reasons that we're monitoring our water quality is to help secure resources for water quality improvements. So now I'm going to take you through the journey of what one of our sampling events looks like. And that journey starts with lots and lots of bottles. Every sampling event actually takes over 200 bottles to collect our samples properly. So from these bottles, we collect water samples for a couple different things. First, we're looking for nutrients. This includes things like total phosphorus and nitrogen, also organophosphate and ammonia. We also look at pH, so how acidic or basic the water is. And then we also monitor E. coli, which is a type of bacteria that can be found in water. And that one's really important for human health. And then finally, we also have volunteers that go out with our YSI meters and actually collect dissolved oxygen measurements at our sites. So all of this is happening every month at 41 different sites. Volunteers are going out onto bridges, they're bringing poles, they're collecting water samples at the sites, and eventually they bring them to our wonderful drop-off coordinators. So here's a picture of Fred. Our drop-off coordinators are located at 4 different spots throughout the watershed to make it a bit easier for our samplers. And then these lovely drop-off coordinators actually drive the samples to our NepRWA office. So here's Judy dropping off some samples this season. And then after they've been dropped off, the environmental fellow, so me and some other volunteers, work to make sure that we've done all of our paperwork properly and that all the bottles are labeled correctly. So that's kind of the story of how we collect those samples. Now we're going to get into some of the data. So I'm going to be talking about a couple specific parameters tonight. These are the ones I already talked about. We're going to be talking about these 3 specifically in terms of data. So I mentioned that we collect a couple different nutrients data. The one I'm going to be focusing on is phosphorus. The main reason for that is that tends to be the most important one at our sites for monitoring. I'm not going to talk about pH a whole lot tonight. All of our sites actually normally fall right within the standard pH, which is a really good thing. That means our sites are healthy for pH. Doesn't make it super exciting to talk about right now. So we're going to skip that for tonight. And then I'm also going to talk about bacteria, so our E. coli levels in the water, and finally dissolved oxygen. Dissolved oxygen is really important because the critters and plants that live in our waters need to be able to access oxygen just like we do. We pull our oxygen from the atmosphere. Critters like fish and macroinvertebrates use their gills to access oxygen in the water. So we can measure the amount of dissolved oxygen in the water and see how much is accessible for the things living there. Also going to talk about phosphorus. Phosphorus is a nutrient that can end up in our waterways, normally via stormwater runoff. When we hear the word nutrients, we normally think that that's a really good thing. But when we have an excessive amount of nutrients in a water body, it can actually lead to some pretty disturbing things, including algal blooms. So this picture right here, it almost looks like a field, but this is actually a pond covered in green algae. And this is due to phosphorus loading, so having too much phosphorus at this site. And then next, we're going to talk about E. coli. And E. coli is a type of bacteria that is present in the intestines of animals and humans. And that means that when any kind of animal or human waste ends up in our water, we can have high levels of E. coli. While most E. coli doesn't actually make us sick, they can be indicators of other things that end up in the water that can make us sick. So this is a really important indicator for human health and whether it's safe for people to actually go in the water. Okay, so before we get to the actual graphs and the data, I want to remind people that the reason we're looking at these numbers is to try to help secure resources for water quality improvements. And the way we actually is by collecting our data, high quality, submit to the state. So we submit every piece of data that we collect to the Massachusetts Department of Environmental Protection. And they actually have different standards that they consider for each of the parameters that we send to them. So X amount of dissolved oxygen is considered healthy, this much isn't considered healthy. And that's how I'm going to be talking about our data for this presentation. So thinking about those categories, we can start with dissolved oxygen. Really healthy areas for dissolved oxygen fall under kind of the gold standard for cold water fish. Cold water fish require a little bit more dissolved oxygen than other fish. And those spots will be marked with our dark green color. And then we have warm water fish who still require a decent amount of dissolved oxygen, but not quite as much. Those will be in light green. Then we have bodies of water that are unhealthy considering their amount of dissolved oxygen. So it makes it difficult for things to live there and be successful while living there. Next, we have phosphorus. This one's a little bit more of a complicated story. Earlier, I was talking about how phosphorus can lead to things like algal blooms. Algae can grow much more easily in still water. It doesn't have to fight the flow of water. And that means that our standards for still water, like ponds and lakes, are much more stringent than those for flowing water. So we want even less total phosphorus in our ponds than we do in our flowing water. But for my graphs, I'm really going to be talking mostly in terms of whether it's healthy for that water body or unhealthy for that water body. And then next we have E. coli. And E. coli is really thought of in terms of human health. So our top quality is levels that are considered safe for swimming. They won't make humans sick. Then we have safe for boating. So there's some E. coli in the water, but we can still recreate on top of it as long as we're not, running around in it. And then there's unsafe for recreation. So E. coli levels are so high that we don't even suggest getting on a boat in the water. So we'll start with our dissolved oxygen results. And I'm going to be using the same graph format throughout the presentation. So I want to make sure everyone has a really good understanding of it. On our x-axis, we have our months that we sampled. They're in order, May through October. On our y-axis, we have our portion of sites meeting each standard. And so each of these bars is equal to 100% of our swim-in sites. And then the portion of that bar in each color is the portion of our sites meeting that standard. So we can see for May, we have a really good start. Almost all of our sites are safe for cold water fish. Another 5% are safe for at least warm water fish. And very few sites were considered unhealthy. We had a couple of sites we actually weren't able to collect measurements for. And I'll give you a second to kind of digest and make observations about this graph. I know something that really sticks out to me is the fact that most of our graph is green, right? So we've got pretty good levels of dissolved oxygen throughout the months at our sites. Hitting about 75% healthy for at least warm water fish throughout the season. So that's a pretty good thing we're excited about. And if you look at all of the samples collected from the 2021 season, 76% of them were considered healthy for fish throughout the season. So that's pretty great. There are times when, say, there are conditions that might make one sampling event not look so great, but in general, we're looking pretty good. These high dissolved oxygen levels are good for the critters that live here. So this map on the left is showing a map of the different brook trout populations in the Neponset watershed. This is something we're really proud of at NepRWA. The Neponset River actually has some of the largest brook trout populations in eastern Massachusetts. And that means that we're providing a nice healthy home for them. So we're happy about that. As well as being healthy for the brook trout, it also means that we've got nice habitat for things like tadpoles to survive, as well as critters like macroinvertebrates or for fish to lay their eggs in. So dissolved oxygen is really important for the animals and critters that live there. Next, we're going to talk about total phosphorus. So this is the same graph that we saw before, just graphing a different parameter. So we've got our months on the x-axis down here, and then the portion of sites meeting each standard on our y-axis. So we can see that we start the season off really well, and then June through September, we're not looking so great, and we come back up in October. I'll note that this graph right here is graphed with our standards for flowing water into ponds. So that standard's more stringent. If we graph our flowing waters looking at the standard for just flowing water, our results look much better. It looks like our ponds are much more healthy in terms of total phosphorus. The reason for this is that a lot of our sites actually lay between those two different standards. So this is a graph showing the average total phosphorus at a site. So we have our different sites listed on the x-axis and the average amount of total phosphorus throughout the 2021 season. And then we've got our two lines here, one for flowing water and one for water that flows into lakes and ponds. And we can see that a majority of our swim-in sites that are at streams and rivers fall right between those two different standards. So our sites, they might not be reaching the standard for flowing into a lake or reservoir, but they're close. That means that we can make improvements and we can get these total phosphorus levels down. So that's a good sign. If we're thinking of things at that more stringent level, only 30% of our sites had a healthy amount of total phosphorus for the season. And this led to a couple different things. We've been looking at this picture of a very green pond here on the left. That's actually Crack Rock Pond in Foxborough. So Crack Rock Pond is notorious for their high phosphorus levels. And that means that they've had many algal blooms, especially this season. There were other locations in the watershed that were also having issues. So Lake Massapog and Sharon had to shut down multiple times because they had unhealthy levels of cyanobacteria. And I include this picture of the storm drain to remind people where phosphorus comes from. A lot of phosphorus can come even just from your car exhaust that eventually ends up running down our storm drains with rain and eventually directly into our streams and lakes. So a reminder that most of our storm drains don't get filtered. They actually just run right into our local waterways. And then next we've got our E. coli results. So again, we've got our months on the bottom axis, the proportion of our swimming sites meeting each standard on the y-axis. We can actually see quite a bit more variability in the E. coli than we saw in our previous two parameters. We start off really well in May with almost all of our sites safe for at least boating. We see a small dip in June, an even bigger dip in July. And we come back up in August, a little dip in September, and then back up in October. So we end and finish really well. And then we have some ups and downs throughout the season. One thing that I really like to mention and make sure people are aware of is that we're only sampling once a month, right? So these bars are representative of a single day in May. It's a snapshot. It's not an average for the entire month. So that's an important thing to keep in mind when you're looking at this data. If we consider all of the samples that were collected in 2021, 76% of them were safe for boating, which is great. 55% of them were safe for swimming and boating. This meant that we ended up having a really great recreational season out on the Neponset River. I started with a video of someone paddling down the Neponset. This is actually a video of a guided tour that we did on the Neponset River in the fall. It was really fun. I think we had 5 different water vessels out. I think we had a paddleboard, a foldable kayak, a canoe, a regular kayak. So people are getting out on the water and having fun, which is always nice to see. In summary, we had really good dissolved oxygen measurements. We're pretty happy about those. Our phosphorus leave room for improvement, but we're close, right? We're right between those two standards. So it means that while we have high levels of phosphorus, we're not blowing it out of the water everywhere. So we can make some improvements and hopefully change those results next year. And then our E. coli is kind of a mixed bag. You know, we had some really great months and some not so great months. But something that I haven't talked a whole lot about during this presentation so far is that context is really important. So I've been showing you these general graphs of our sites throughout the Neponset watershed, but our watershed is pretty big and it experiences lots of different things throughout it. So one important piece of context is location. Here's another map of the Neponset River watershed. Each of these spots indicates a swim-in sampling site, and we're using the same color coding that we've used before. So the dark green spots are safe for swimming, the light green are safe for boating, and the yellow are unsafe for recreation. And this was an average of the E. coli measurements taken throughout the season. So some observations that we can make. We can see that it varies by location. For instance, we tend to have healthier E. coli levels here towards the headwaters than we do out towards Boston. Some different things we can think about when we're looking at these maps and trying to interpret why we see different levels in different locations is population. So we have a whole lot more people living over here by the mouth of the river than we do at our headwaters. That means that there's more infrastructure, more potential for poor pipe connections or cracks in pipes that lead to sewer overflows. Another thing we can think about is impervious surface cover. So more people living up here, we probably have more streets and more roads. Those impervious surfaces lead to things like stormwater runoff, which can include dog poop and bird poop and bring E. coli right into our waters. And then another piece of the puzzle is the fact that different towns have different resources to fix issues or even look for issues. So lots of things to think about when we're looking at our data. Another really important piece of context is the fact that we had a really, really wet year. July 2021 was officially considered the wettest Massachusetts July in history. By September, we actually had more rain than we'd had in all of 2020. So lots of rain coming down this year, and that was definitely seen in our water quality. So I've made us two graphs here, one looking at E. coli in our dry events and our wet sampling events, and one looking at total phosphorus in our dry and wet sampling events. So a wet sampling event is a sampling event that had more than 0.1 inches of rain in the 72 hours preceding. So basically we had rainfall before we went out and sampled. And our dry sampling events are sampling events that didn't have rain before the sampling. And we can see that our dry sampling events have great results. Almost 100% of our sites are safe for at least boating, and most of our sites are even safe for swimming. But then when we look at our wet sites, those numbers go down drastically. So only about 50% of our sites are safe to swim in after a rain event. One thing to think about with this is that we can mitigate our own risks of swimming by thinking about what the weather has been like in the 72 hours preceding our adventure out into the water. So if you're thinking about swimming, if it's rained in the past 24 hours, maybe think about a different way to get outside and enjoy the weather. The other thing that we can think about is the fact that E. coli gets flushed out of systems pretty quickly. So rain events bring a lot of E. coli into our water, but when it's dry, the E. coli is able to flush out of the system and we don't see it persisting as much. Whereas if we look at our total phosphorus results, we can see that there isn't a big difference between our dry and wet levels. And part of that is because phosphorus is really persistent in a water body. It gets taken up by things like algae really quickly, it gets held in that system, and it takes a much longer time for it to cycle out of an area than something like E. coli does. So that means that if we have a big rain event at the beginning of the season, we might still see high phosphorus levels 2 weeks later. The reason I bring up rainfall as something we should be thinking about is because with climate change, Massachusetts is going to be seeing more and more extreme precipitation events. So this was a graph that was made by the federal government, and they were basically looking at different climate change trends in each of the states that we have. And the thing that they noted for Massachusetts was that we're going to be seeing more and more of these extreme precipitation events. So we can see a pretty clear trend here of an increase in those. Which means that these issues with water quality that accompany our extreme precipitation events might become more and more common too. Okay, so now we're ready to talk about our future plans. I'm gonna start with our swim-in plans. Right now we're in winter, so we're thinking about our next season, which is very exciting. We're thinking about adding a few more sites, potentially at some more lakes and ponds, which is great because we definitely only have a couple of those. It'll be great to add some more diversity. We're also talking about adding some continuous data. So one of the things that I mentioned with those graphs was that we are really just looking at a snapshot of that month. We're looking at one piece of data for that entire month. We're not looking at an average or continuous data. So by adding things like dissolved oxygen loggers or temperature loggers at a few of our swim-in sites, we'll be able to get a more holistic view of what's actually happening throughout the season. Another thing we always try to do is follow up on hotspot monitoring. So if we notice the site is having particularly bad results for a certain parameter, we try to go out and investigate what caused those. So those were some of our swim-in specific future plans. Now I also want to touch on some of the other exciting news happening in the Neponset River watershed. Some of these are projects that NepRWA has been really heavily involved in, and other ones are things that we're just excited to see happening in our own watershed. So we'll start out in BMP. So a BMP is a best management practice, basically looking at ways that we can deal with stormwater that's been polluted in a more efficient way than having storm drains just bringing it right into our streams and rivers. So this here is what's called an infiltration basin. We can see a big depression in the ground. And basically this allows water that's collected on streets and roads during storms to collect here and more naturally infiltrate into the ground. So rather than the water being brought up by a storm drain and going into a stream, the water is able to naturally flow down into the groundwater. It gets filtered by the soils. And this one is actually going to have a really positive impact on one of our streams in the watershed, Beaver Meadow Brook. We do have a swim-in site, so we're excited to see what the results look like. It's been projected that annually this BMP will help to prevent 60 pounds of nitrogen and 8 pounds of phosphorus from entering the stream. So this is really exciting and big news for water quality. Another project we're working on, which I mentioned earlier, is the Mill Pond Dam removal. That was the drone footage you were seeing of the pond. This is the actual dam that we're working on removing. And here's a picture with the flash boards removed. So really exciting. We're already seeing a better flow of the water. Eventually, I think maybe next week, we're actually going to be taking the dam out, which is super exciting. This will help open up the river, have it flowing more naturally, and allow critters like brook trout to migrate throughout the stretch of river more easily. We also just heard news that Foxborough applied for and was given a grant to look at the feasibility of removing the dam at Crack Rock Pond. This is really, really exciting. This is the pond that I keep talking about with the phosphorus issues that lead to algal blooms. So if we're able to eventually get rid of this dam, that'll make it so the flow of the water is stronger and it's much easier to prevent algal blooms and have phosphorus flow through rather than getting trapped in the pond. And then kind of on a more broad perspective, there's actually some plans to get part of the Neponset River listed as a Superfund site. So right now it's been proposed. We're hoping it might get listed this spring. It's about 3.7 miles of the Neponset River where it's been identified that there's lots of PCBs in the sediment. So not actually in the water itself. The water is pretty safe to be in, but the sediment or the ground underneath the water has been found to have high levels of a chemical that's been shown to cause some health impacts in humans. This is really, really exciting because it potentially is going to bring a whole ton of federal money into our watershed to help clean our river and make it an even healthier place for us and the wildlife. So we're pretty excited about this. Keep your ears open because hopefully we'll be talking a little bit more about it this spring. And I would be a horrible coordinator if I didn't at least acknowledge the swim volunteers and their amazing work once. So I want to say a huge, huge thank you to all the swim volunteers who give us 3 very early hours every month in the summer to help collect these water samples. None of this work would be possible without you all. I so appreciate it. It's been a pleasure, and I'm looking forward to starting off the 2022 season with you guys. So that date is approaching quickly. I'm getting a little anxious about all the things we need to do. So you'll be hearing from me in your email pretty soon. And I think with that, I'm happy to answer any questions. So the way we are thinking this could work is that people can put their questions in the chat, and then Sean will actually read them out to me and I can hopefully answer them. I've also put my email here on the screen. Please feel free to reach out with any other questions or comments. I love hearing from folks, and especially if you're interested in a specific site or swim-in location that we have.
All right, so we have one question already, which is about Pine Tree Brook and whether the E. coli levels there are particularly bad. This may be it's hard to interpret. So the question is about the source of Pine Tree Brook being in Blue Hills and why E. coli levels might be high at the first site on Pine Tree Brook that we sample.
Ooh, that's a great question. So I talked a little bit about how context is a big factor. So even if water is coming from a really healthy place like the Blue Hills, there might be other aspects that might be playing into the water quality there. So it could be like a leaky sewer system that is leaching into the pond. Let's see. And I'm also happy to defer if Sean has any other specific things he might have observed at Pine Tree Brook. But if you email me, I'm happy to share some of the specific data for the site as well.
Yeah, and I think, sorry, keep scrolling back to the bottom every time. Yeah, it's because I think Tabor had mentioned it's the first samplings that we have is below Pope's Pond, and that might be a big influence. Let's see what other questions we've got.
It looks like we have a couple questions about specific sites. So I'm also going to put this on the background if people are interested. This graph is showing the average E. coli measurements from sites for the 2021 season. And you can see that I put the different standards on here. The green is safe for swimming and the yellow is safe for boating. And I'll say that a lot of these sites that have really high averages are being heavily influenced by July, when we had a really crazy rain event right before our sampling.
Yeah, and I would also say, Megan, you can point people to the interactive website. Even though it doesn't have this year's data up there yet, it will eventually.
Yeah, great point, John.
All right. So I have a question. Would we assume high phosphorus levels in June through September has to do with people spreading fertilizer pellets on their lawn?
Yeah, so a lot of people think that phosphorus issues are specifically coming from fertilizers. There's actually a rule in Massachusetts that says that you're not allowed to, I believe it says that you're not allowed to apply fertilizers with phosphorus unless your yard has specifically been tested and come back as needing more phosphorus for your lawn or whatnot to be healthy. So I think fertilizers are regulated to have whether they have phosphorus in them or not. And they're supposed to be regulated in who they're sold to as well. A lot of the phosphorus can actually come from things just like car exhaust. So areas with lots of commuters and cars can have phosphorus issues in their waterways.
All right, great. Okay, scroll down. So there's a question here about what is the criteria to remove a dam. I think maybe I should just answer that one briefly. So it depends a little bit on the size of the dam. Some, a dam that is essentially loose rock constructed in a stream can be dismantled pretty easily and without too much oversight. But bigger dams need to be evaluated by various state agencies. And I don't want to get into too much exactly how that works, but it depends a little bit on the size. And this question about the basis of the collection and drop-off sites for swim-in. Is that available anywhere online, Megan, that you can point them towards, or we can just send that out?
In terms of where our sampling sites are or where our drop-off sites are?
I think basically how a typical sampling day works.
Yeah, we have some information on our website for sure. If you have specific questions, I'm also happy to answer. Basically, the 41 sites are spread out throughout our watershed, and we have 4 different drop-off locations in different towns. We have one at the NepRWA office, we have one in Milton, one in Walpole, and one in Norwood, maybe? I'd have to look at the map, but I'm happy to provide that information to anyone who's wondering.
Okay, and we'll go to this one. Someone asked whether COVID has been found in any of the ponds in our watershed.
Ooh, that's a good question, and not something we test for. So I actually don't have any specific information on that. I don't know if Sean, you've heard anything about that.
Yeah, I'll chime in and say I don't know if anyone's tested for it specifically, but if you are concerned about it, COVID, when they talk about COVID and wastewater, it's the genetic signature of it. It's not infectious particles. So it's just really a measure of how much, the population is experiencing the virus and not anything infectious in the wastewater. But they also primarily would test the wastewater because hopefully most humans are using the wastewater system and not the stormwater system. Okay. There's a question, can algae pollute the water?
So algae, phosphorus is considered the pollutant, and then algae uses that pollutant to grow. And basically, phosphorus is something that plants are constantly looking for, and algae is really good at picking it up and using it really quickly. And in the process of using that resource, phosphorus, they also use up other resources in the pond as well, including things like dissolved oxygen. So when there's high levels of algae in a water body, it can make it really difficult for other things to live there. There's also issues with human health. So cyanobacteria can make people really sick. And that's actually why we had things like the Lake Massapog get shut down multiple times this summer, was because of the cyanobacteria levels.
And then we have a question whether there should be testing for coli. Megan, do you know anything about that?
I'm sorry, you broke up for just a second.
Oh, sorry. About a question about whether it's better to test for coliforms, fecal coliforms in the water, or E. coli itself. I don't know if you have any thoughts on that.
So E. coli is a type of coliform, and basically you can test for generally all coliforms, or you can test more specifically for E. coli. And E. coli is going to be lower than your coliform because it's a type of coliform. So we do some testing in-house on E. coli levels in different water bodies. And we will look at coliform and E. coli, but the thing that we're really focused on is the E. coli.
Yeah. Oh, here comes the question. How does plastic litter affect the water? And do we have any information on that?
Love that question. I'm just in the process of organizing our spring Neponset River cleanup. And we definitely see plastic litter in our waterways. And this is bad for multiple reasons. Plastics can leach chemicals into our water, which are bad. It can also impact things like wildlife. They can sometimes think that plastic is food and eat it. And then they think they're full and they'll stop eating the things that they need to eat in order to survive. So plastic is definitely an issue in the Neponset River, just like it is in almost any water body in the world. But we're trying to do some things to help lower that influence. So if you're interested, April 23rd is the date of our spring cleanup, and we love to see as many people as possible out there cleaning up our waterways.
And there's a question here, which water is best for us? I'm not entirely sure. Michelle, if you want to clarify that question, we'll come back to it. But do you know, Megan, if there are any visible spring waters flowing in the Neponset watershed? I don't know the answer for that.
Yeah, I don't know if anyone else from NepRWA has an answer for that, but I do not have a spot off the top of my mind.
All right. You can wait around for a few more questions or clarifications, but I think that's it.
Great. Okay, well then I'll hand it over to you, Sean, and I will
so
questions for you.
Oh, someone's saying spring water or faucet for which water is best. I don't know if you have a point.
Theoretically, all the water coming out of our taps should be tested if you're getting town water, and tested and treated to make sure that it's healthy to drink. Spring water is thought to be really clean, but I also would be hesitant to tell anyone to go out and drink water that hasn't been tested.
All right. So let me go ahead and...
It looks like Ian provided an answer. Ian is the executive director at the Neponset River Watershed. He says, yes, there are some visible surface springs around, though most of our springs come up right at the edges or bottoms of rivers and lakes, so you don't really see them.
Great. All right, so I will take over the screen sharing. All right. So hi, everyone. I just wanted to give a brief introduction to myself. So I'm the Neponset River Watershed Association's new River Restoration Director. And I started this December, taking over for Jenny Rogers. So just a little bit of background about myself. I'm a biologist and environmental scientist by training, and I just recently got my PhD from UMass Boston. And so my role here at NepRWA really kind of breaks into two major buckets. The first is working with restoration, so identifying sites, working with towns and funding agencies to get restoration projects off the ground, and shepherding through the process. So as Megan mentioned, we're working currently on the mill pond over in Norwood, which is underway, and then hopefully, identifying more projects in the future and finding the funds and getting those projects underway and completed. On the other side, the water monitoring side, which everyone is here today to hear about is overseeing the water quality through the swim-in program alongside our environmental fellow, which this year is Megan. So because I'm new, I kind of wanted to give a little bit of background about myself and talk a little bit about the most recent research I've done in my doctoral work. So as I said, I just recently got my PhD in environmental biology at UMass Boston. And we really, in doing that, the question I really wanted to ask is how aquatic systems respond to restoration. So a lot of times when we're thinking about water quality, we think about what's the negative impact we have on the waterways. What way are we polluting them? How do we control the E. coli, the phosphorus that we see in our systems? So the kind of reverse question is, what can we do to make those waterways better? And how do they respond to our efforts? So the kind of major context for this is cranberry bog restoration. So cranberry farming is a huge form of agriculture here in Massachusetts. And in the last several decades, growers have faced enormous difficulty in turning a profit. There's cheaper cranberries coming from Wisconsin. Land here is expensive, and many farmers are aging out of the system, and they're unable to sell the land to someone who wants to maintain it as a farm. So conservation and restoration is seen as an exit strategy. It's a way that the government and municipalities can get these lands back, put them to good use in bringing back natural wetlands or natural flowing water systems, and regain some of that that missing ecosystem function. So specifically in the sorry, these things are popping up. Megan, can you handle the people coming in? So specifically for the project I was working on, this is was Tidmarsh Farms down in Plymouth, Massachusetts, which is a former cranberry bog that underwent restoration in 2015. So the real question is, how do we take it from what it looks like here? You can see this culvert running through a very simplified channel, and move it to a much more natural, wild and flowing river system. So luckily, we were able to partner with Mass DER to collect our pre and post restoration data and really understand what happens in that first couple of years after restoration. So one thing to think about restoration is not simply, something is damaged and we come in and fix it, and then it's immediately restored. There's an intermediate step, which is the construction phase. And as you can see, if you've seen any construction projects alongside the river anywhere, it's a harsh environment that's created. And we would call this an ecological disturbance. And so it makes it a little bit difficult to detect the benefit we get out of it, because we're also damaging the system in order to fix it a little bit. And hopefully the end goal is, again, that nice flowing wild system. So when we're looking into restoration projects, we also want to be aware of that disturbance event of the land being moved around and all that soil being mobilized, and really take that into account. So the question is, what do we care about in these systems? And coming from my bias as a freshwater researcher, when I designed this experiment, I was interested in a couple of things. The first is physical stream characteristics. So its length, its width, its depth, the amount of woody debris and cobble for fish to play around in. We sampled that. The next one, water chemistry and quality, very similar to the swim program. We take water samples. Instead of taking it monthly, we took it seasonally, but very similar idea. Brought it back to the lab and investigated it. And then we looked into aquatic insects. As Megan mentioned, they are some of the most diverse things that live within our stream systems, and they need that oxygen. They need those resources to survive. Next, we also wanted to look at the fish population. So aquatic insects are great. They tell us a lot about what's happening in the moment, because they have pretty short lifespans. They can tell you what's happening that year. Fish tend to be a little bit more longer lived. They tend to travel a little bit more. They can tell you a little bit more about the system as a whole over the several years. And then finally, we want to know a little bit about the landscape level impacts. So streams don't sit quietly in their banks. They expand out to their floodplains, and they affect the land use around their watershed. So we want to look at all these features and kind of see how the system responds to restoration. And just to kind of cut to the chase here a little bit, what we kind of found is that we found some improvement in the physical features of the stream. We brought in land movers, we rechannelized the stream, we dug out some expanded sediments and kind of opened that up, connected it back to the floodplain a little bit, and improved the physical features there. The areas where we found a kind of bounce back effect, so remember I mentioned that kind of disturbance of the restoration process, we found that water chemistry, the aquatic insects, and the fish populations, they all suffered an initial decline, which we kind of expected, but they bounced back within 1 year. So that's pretty great news. It means that here in Massachusetts, even doing kind of fairly intensive active restoration, we can expect these systems to bounce back pretty quickly. And then kind of more ambiguous results. These landscape level impacts, they're the ones that are going to take much longer to show any kind of significant impact, right? As we change the land use, it requires trees to grow up, and it's going to be a little bit harder to detect in only a few years. Okay, so what does this all mean? So this has a couple of implications for near-term change, but really what it's doing for us is providing feedback to restoration practitioners. And part of this is that restoration projects, both in Massachusetts and everywhere else, they tend to happen in small quantities, one after another, right? No one has billions of dollars to put towards this right at this moment in one location. We can't just restore all the streams at once. So what that means is that we can learn from each one and make the next one better. So that gives us a little bit of a framework to work with. We can plan a restoration. When we do the restoration, then based on what happened in that project, we learn a little bit more for the next time, maybe make it a little bit better. It also gives us a little bit of opportunity for continuing our research and education. And in particular, this project down at Tidmarsh Farms worked alongside something called Living Observatory. So Living Observatory is an umbrella organization that was created out of this restoration project, bringing together academics, state and federal researchers, and nonprofits to set up the new Tidmarsh Wildlife Sanctuary, which is owned by Mass Audubon, as a perpetual research and education center. So the project looking at the restoration that I did is going to continue there for many years to come. They're going to couple it together with more restoration projects as they come on board through Mass DER, and they'll be teaching school classes and interested members of the public about this for the foreseeable future, which is a really great outcome to come out of this. And so what kind of lessons can we take away from this for here in the Neponset watershed? One kind of thing that I think should really take away from this is to have clear goals for restorations. So it's a kind of an umbrella term for restoration, but every restoration is unique, right? So if you are addressing something specific like a dam, the restoration has to be geared towards that. It has to be addressing the changes or negative impacts of that specific impairment. So impoundment, moving the sediment that's behind it, rechannelizing the channel itself, all go alongside of a dam. We want to know if there's specific species we care about. So again, at the Mill Pond, we're interested in brook trout. That means we have to bring back specific habitat and specific cold water features that they require. If it's a stream that doesn't have brook trout on them, maybe your goals are a little bit different. But in either case, we want to ensure there's also adequate monitoring, right? So one of the things we can take away is that you can't know what happened to a restoration the next day. It's something you have to find out over the next several years. You want to make sure that you're there, collecting that data, doing the work that Swimmin advocates of that long-term data and helps us understand what's changed. And then finally, we want to make sure we incorporate everything we've learned from projects, not just here, but across the New England region and even across the nation. So we know what to do and learn the lessons that others have learned too. And so that's kind of it. I wanted to keep it quick and for you all, and I'm happy to take any questions that you have, and I'm excited to meet you all when you come to start the swimming program this year, or start the sampling season this year. Thanks.
Okay, Sean. It looks like I have one question in the chat so far. Someone's wondering if we found any PFAS in the river, or if there's any information we could give them on PFAS in the Neponset watershed.
Yeah, so we don't test for PFAS, and I know that. There has been some talk about whether we should be testing for it, and that's something we're looking into in the next several years. But at the moment, the standards for testing it and being able to submit that data to the state aren't there yet, so we haven't really taken advantage of it.
Oh, and then Ian asked, can you explain what PFAS is?
Can I explain what PFAS is? Yes, it is poly and per fluorinated alkyl substances, I think.
Pop quiz.
Is a pop quiz. But they are basically persistent organic chemicals that are bound to a fluorine molecule. I kind of get a little technical, but they're things like Teflon and similar chemicals. Hope that was right. Ian will surely correct me if I'm wrong.
Well, and Ian also mentioned that there's been extensive testing of PFAS in drinking water by Mass DEP.
Yeah. All right.
Great. Okay, I think that's all we have for tonight. Thank you everyone who joined. This has been great. I hope that you learned something or enjoyed it, got to get in touch with the water quality in the Neponset watershed. And again, if you have any questions, please feel free to send them our way. You can find Sean and I's email on the Neponset website, and I'll pop mine in the chat right now as well.
Bye, everyone. Thanks.